Okay, sorry about that, but we ran out of time. 50 minutes is my limit, so I had to upload that one to YouTube, and we'll start the second half. So, lipids. We talked about carbohydrates and proteins, and the other major organic group, of course, are lipids. These are non-polar hydrophobic. I think I said that, didn't I, in the last one, where, as we know, oils and water don't mix terribly well. Of course, they're a wonderful store of energy, and adipose tissue is essentially lipids stored in cells for later use if needed. The cell membrane, of course, is made of two lipids, a phosphobilipid layer, and steroids such as hormones and vitamins are based on lipids. So lipids are critically important to us, and they contain two parts, like triglyceride and the fatty acid. Sort of lost my way a bit there. Okay, so membranes. The important part of our first sac. So we can see here on this diagram the two phospholipid layers with the hydrophobic heads and the hydrophilic region in between where the tails meet. So it's very difficult for water-soluble things to cross the structure of this oily material. Of course, that's why we find these protein channels. Any cholesterol marked on that one? No. So we tend to call this a fluid mosaic model of the membrane, where the two sides, the two layers of phospholipid move across each other, and embedded through it to try and cause some linkage is cholesterol and all these proteins and carbohydrates, or glycoproteins, so mixtures of carbohydrate and protein, which all have a role to play. So some of them are self-markers, others are docking stations for proteins, others are protein channels, and the membrane's covered in so many things that it allows us to communicate with other cells and to communicate inside the cell, and to essentially control what comes in and goes out of the cell. And of course we're going to look at how water apparently moves by osmosis across this. Diffusion, simplest form of movement of anything. So it's the movement of molecules of a substance from an area of high concentration of that substance to an area of low concentration. Now clearly gases is quite easily. Most of the stuff we need needs to be dissolved in water, so that we've got areas of high concentration moving to low concentration. Requires no energy. The materials are going to move simply by themselves. As long as they're small enough and lipid soluble, they'll cross our membranes. Osmosis is essentially a form of diffusion, but it applies simply to water and water only. And the water moves in relation to the concentration of the solute dissolved within it. So in our little diagram there, the green is the solute, so it could be, let's say, sugar, and little blue dots are water, and little blue dots will move across that membrane from area of low concentration of the solute to the area of the high concentration of solute, which will then, of course, balance out. Sorry about that, we've just had an interruption from home. And we talk about substances being in an isotonic situation, a hypotonic situation, and a hypertonic situation. And we'll look at those in a bit more detail now. So, isotonic, we've got equal concentrations of water and solute either side of our membrane. So that yellow blob in the middle of this diagram could be a cell, or a cell vacuole maybe. Things don't stop moving, but movement is balanced, so that essentially we're looking at equal concentrations either side of the membrane at all times, but things are in movement. We tend to think of equilibrium being a sort of static process, but it remains fluid. Hypertonic, so in this case we can see that inside of our cell we've got lots of water but not very much solute. So in relation to its environment, the cell is hypertonic, and the water will leave the cell, or the vacuole, and cross the membrane to that high concentration of solute. And of course this will cause that cell to dry out, or that vacuole to dry out. And either the cell becomes desiccated, or the vacuole becomes flaccid, and the whole cell becomes flaccid. The opposite is hypotonic, And so in this case we've got lots of water molecules outside of our vacuole or our cell, and they move in because of the concentration of materials inside the vacuole or cell. And then in a plant, this would cause the vacuole to swell, and that would cause turgor and a nice strong plant standing tall. In an animal cell, this could actually cause a rupture of that cell because there's no cell wall to stop the cell bursting. And you want to be able to control that water coming in and balance it by getting materials out as well. We'll look at all these sort of things because this is how a plant opens and closes its guard cells around its stomata, of course, by adjusting the turgor of the vacuoles in the guard cells. And of course, there are many ways to move things across the membrane. So we've looked at diffusion and osmosis. These are both energy-free and move all by themselves because they can. But we also have what we call protein-mediated transport. So facilitated diffusion is simply a protein channel which hosts a particular solute. That solute may not be small enough to cross the membrane by diffusion. It may not be lipid soluble, so it can't cross the membrane, such as glucose. So glucose has its own protein channel. It still moves from areas of high glucose to low glucose, but it requires a channel to be open for it to be able to do that. And the cell has the ability to open and close those channels if it needs to. No energy is needed. Again, it's just using diffusion. Active transport. Now, active transport again uses these protein channels, but it requires energy to set up a concentration gradient. And the sodium-potassium pump is a classic example of an active transport situation. So we're moving potassium and sodium in relation to each other to move materials into or out of the cell, so across that membrane. This does require energy though. And both of these are faster than just simple diffusion because the channels allow you to make things more effective. I've got some animations there, we won't bother with those right now, and I've finished that PowerPoint finally. This is part 2 of chapter 1. See you guys in class. Bye.